Hello everyone and welcome to Chapter 1A, Microbes Among Us. So let's jump right in. Our focus of study in this class will be microbes. There's two terms we have to start off with. First off, microorganisms. These are living organisms that are too small to be seen with the unaided eye, meaning we're going to have to use an extension of the senses, like a microscope. This includes all of the green highlighted words at the bottom of the slide. Bacteria, archaea, fungi, algae, protozoa, and multicellular parasites. They are cellular organisms that are alive. Microbes, on the other hand, can be living or non-living, meaning they do include all of the green highlighted words, but they also include non-living infectious agents, like viruses and prions. So where are these microbes found? Simply, everywhere. Any place you can possibly think of, and some places you might not. They are ubiquitous on Earth, meaning you can find them on any surface, living or non-living. They populate the air, the water, the soil, and have evolved intimate relationships with plants and animals. Without microbes, life on Earth would cease. This is due mainly to the essential roles microbes play in the systems that support life on Earth, like nutrient cycling and photosynthesis. Also, the physiology, nutrition, and protection of plants and animals, including humans, is dependent on various relationships with their microbes. This picture from the previous slide is Pseudomonas aeruginosa bacteria found on a household appliance. This is what's found on 1 square centimeter of a fridge handle. This photo is bacteria on a human foot. This is the bacteria and microbes found on the surface of a cat tongue. This is bacteria from a train door handle, a very common place where we find microbes on a moist surface. This is bacteria on the human tongue and cheek cells. This is a single colony of bacteria from a 4-day-old baby's gut. Bacteria, again, are everywhere. And surprisingly, there are actually more bacteria in a human body than human cells. A 154-pound male, a height about 5'7", contains approximately 39 trillion bacterial cells, compared to only 30 trillion human cells. This is a ratio of about 1.3 to 1. And these microbes are usually about 3% of body weight. So in the male expressed above, he has about 4.5 pounds of microbes. Most of these are found on the gut. The picture on the bottom left of this slide is actually intestinal bacteria. Those green dots, that's Staphylococcus aureus. The picture to the right shows all the places we find the most bacteria in the body. This includes areas like the forehead, the skin, the ear, nostrils, armpit, elbows, and inside the body, around some of the organ systems. Generally, the only places that we don't have bacteria are places that remain sterile. These include the blood and the cerebrospinal fluid. Clearly, the human body is colonized by a vast number of microbes. There's actually a term for this. It's called the human microbiota, although you may hear the term microbial flora as well, since bacteria were once classified as plants. The majority of these do live in our gut, particularly in the large intestine, but there are many on other surfaces that also have helpful functions, including the digestion of food, the regulation of the immune system, protection against other bacteria that cause disease, and the production of vitamins including B12, thiamine and riboflavin, and vitamin K, which are integral to the production of blood coagulants. The link between these microbes and our health is the focus of a growing number of research initiatives, and new insights are emerging rapidly. These are several examples of what we're learning about the human microbiome. The gut microbiome is different between obese and lean twins. Obese twins actually have a lower diversity of bacteria and higher levels of enzymes. This means that obese twins are more efficient at digesting food and harvesting calories, which leads to weight gain. Obesity has also been associated with a poor combination of microbes in the gut. Another example of this is type 1 diabetes. This is an autoimmune disease associated with a less diverse gut microbiome. In animal studies, bacteria actually play a role in developing diabetes as much as genetics does. A third example, dust from homes with dogs may reduce the immune response to allergens and other asthma triggers by changing the composition of the gut microbiome. It's been found that infants who live in homes with dogs have been found to be less likely to develop childhood allergies. In example 4, fecal microbiota transplantation, FMT, also known as poop transplants. These are clinical procedures that restore healthy bacteria in the colon by introducing stool via colonoscopy or enema from a healthy human donor. This is being used in a lot of different treatments, including treating fatal Clostridium difficile infections and to treat colitis, constipation, irritable bowel syndrome, diabetes, and even autism. The microbiota of humans is vast, but their microbiome is even greater. The microbiome is the genetic material of all the microbes, bacteria, fungi, protozoa, and viruses that live on, in, and around the human body. The number of genes in all of the microbes in one person's microbiome is 200 times the number of genes in the human genome. So we are mostly genes of microbes. The Human Microbiome Project, or HMP, studies the human as a superorganism composed of non-human and human cells, with the goal of describing the human microbiome and analyzing its role in human health and disease. They've even figured out that microbes may play a part in neural regulation. Now that we know what microbes are and where to find them, we have to figure out how to see them. Most microbes are microscopic, so we're going to need an extension of the senses in order to view them. This is usually done using a microscope. So if you look at the diagram below, you can see that bacteria and viruses are usually in the micrometer to nanometer range, very small. Whereas animal cells and plant cells are much larger, sometimes 100 to 1,000 times larger. We are going to be using a compound light microscope in lab, but a lot of the image we'll be seeing in lecture will come from an electron microscope. So what's the difference between these two pieces of equipment? We'll start with the compound light microscope that's on the left. The light microscope is easy to carry and produces color images as long as we provide contrast. That's why in lab we'll be making smears and staining them, so we have a colored cell or background. Light microscopes have less resolution, which means it's more difficult to see different areas of the image, and it has less magnification. Our microscopes will magnify times 1,000. The picture on the upper left-hand corner shows a radiolarian under a light microscope. As you can see, depending on the angle of the organism, you have different areas that have a little bit of blur, so it's not as crisp and clear. When you move to the right side of the screen, you'll notice the image is very, very clear, and you can see all the distinct parts of the organism. This is the radiolarian under the electron microscope. Electron microscopes are very cumbersome. Usually, they weigh hundreds to thousands of pounds and cannot be moved. And they produce black and white images that must be colorized. The pros, they have better resolution, so you can see different points on the image. and much higher magnifications, sometimes times 1,000 to 10,000 magnifications.